Welcome to the Compendium of Knowledge, a living library of ideas, values, and insights meant to spark curiosity, open our minds, and help us see the world with greater depth and humanity. Today, we are joined by Dr. Martin Chalfie, who received the 2008 Nobel Prize in Chemistry alongside Osamu Shimomura and Roger Sien. Dr. Chalfie helped give the world one of the most transformative tools in modern biology: green fluorescent protein, or GFP, a naturally glowing protein first found in jellyfish. His pioneering work showed scientists that you could attach this tiny glowing marker to almost any gene inside a living organism and suddenly watch life from the inside. Instead of guessing what cells were doing, researchers could now see. They could track how neurons fire, how cancer spreads, how embryos develop, all in real time. Today, Dr. Chalfie is a professor at Columbia University, where he continues to explore the mysteries of touch, sensation, and how even the simplest organisms can teach us something profound beyond ourselves. Beyond his research, he's been a passionate advocate for openness in science, experimentation, and the idea that mistakes, even big ones, can lead to the discoveries that change the world. Dr. Chalfie, it is a privilege to welcome you to the Compendium of Knowledge.
Thank you, Aaron. It's nice to be here.
So, firstly, for a young audience, could you please explain in simple terms what you discovered about GFP, the glowing protein, that led to your Nobel Prize, and why that discovery changed the way we study and understand living things?
Well, I'm going to show some images a little bit later, but the basic idea is one day I was listening to a seminar when I heard about this protein. It's a protein that is fluorescent. There are minerals that are fluorescent, other chemicals that are fluorescent, but this is a protein. That is, it's made up only of amino acids. It could be encoded by DNA, and you don't need anything else to make it work. And what I mean by make it work is if you shine, in this case, blue light, you don't see the reflection of blue light. Instead, the molecule changes the blue light to green, and you see the green light coming out. So wherever the molecule is, you will see it as green. And that conversion, that's fluorescence, is a way of being able to look at where the molecule is. And I realized when I was listening to this seminar about this molecule, that it would be an extremely exciting tool to use, because we could have organisms make it in their cells, and whichever cells made it, we would be able to see it. And so the basic idea was, or basic question was, does it work? Could you put it in any cell you wanted and be able to see that cell simply by shining blue light and seeing green? And so I didn't discover the molecule. That was Osamu Shimomura who did that. And he did it when I was still in high school. He did it in 1962. But I realized that it would be a wonderful tool, and my lab showed that we could put this into cells, bacterial cells, nerve cells, various types of cells, and we would be able to see them. And not only see them in fixed or prepared samples, but actually in living organisms. And that turned out to be a very useful tool that many, many people used. And so the Nobel was really about the molecule. And Shimomura, Chen, and I went along for the ride.
Wow, that's very interesting. Thank you. And for the listeners out there, of course, you are viewed currently as a role model and have had a very fulfilling life. But now I begin to wonder what originally motivated you to explore chemistry and the inner workings of living organisms. if not for a Nobel Prize?
So I don't think anyone goes after a Nobel Prize because I think that's almost impossible to do and unlikely. Uh, but I've always been someone that was interested in science. I enjoyed reading science books when I was a kid. I enjoyed taking science courses and math courses when I was in high school and in college. I wasn't very good at it, though. I went and tried to work in a lab, and that did not work out well. In fact, it was such a disaster that I decided when I was about to leave college, I should never work in a lab again. And that should be the end of it. Even though I enjoyed it, I love the ideas. I love thinking about the problems. But I, I had a very incorrect way of thinking about science when I was in school. And that was that true scientists, whoever the world that was, were people that were able to do this on their own. They never asked for help. They never asked questions of other people. And they just somehow innately were able to get it to work, things to work. They didn't work that way. And when I started finally, after I had left college, being able to have the confidence to talk to people and work things out and interact with people, that turned out to be really exciting. So to me, getting back into science had really two parts to it. One was being excited again about the work. And often that's connected with, did it work or not? Did did the experiment actually tell you something? And it did. When I was able to finally get back into a lab, I was fortunate enough to work on a project that worked, and I got excited about the success. And it gave me a little bit of confidence to maybe think, maybe I should do this more and I should go to graduate school. So I went through a period where I was convinced I should never be a scientist. And then the success and support of other people uh, helped me decide maybe I should do this. Maybe this would be a good, fun thing to do. But my interest in science, I think, continues in terms of the, for me, more the excitement of the ideas. Can we figure things out? Can we test our ideas? And what comes of that? And to go off into maybe a slightly different direction, I think of myself not as a chemist. I think of myself as a geneticist. I look for mutants that are defective in various activities. You mentioned before mechanosensation, the sense of touch. We have mutants that are defective in touch. And the interesting thing about genetics, when you go after mutants, variants that are not like the wild type, the normal, and then you clone the genes, you usually find out that you're working on a gene that you know nothing about. That it's a part of biology that's completely new. And to me, starting over and learning something new is really wonderful. It's very exciting. You're suddenly thinking about problems that you never thought before, or I never thought before, and then trying to work through them, trying to understand why something is happening. So genetics is the organism telling you what's important for it, not you telling the organism what is important, because you never know what the gene is until you finally clone it and start studying its product.
That's very interesting. Thank you. And I really admire how you make knowledge into something that should be enjoyed and not something which is something that should be forced upon. And in your explanation, you have previously spoken about how asking central questions and being honest with yourself are in science. So what is this mindset about feeling free and feeling honest to ask questions and feeling happy with not knowing everything? What has this mindset taught you about life?
Well, we don't know everything, so I don't think that's, you don't have to convince yourself you don't know everything. But I I think it's I sometimes when I give talks, I talk about general approaches to science. And I think that there are two things that I have found useful, other people have found useful. The first is noticing the questions that are actually all around us, questions that we may never have thought about before. So I'll give you one example of what I'm talking about. And it's something that people know about and have known about their entire lives. But I have found very few people to have ever asked a question about it. So here's the question. The very first uh, paper I ever had in science, which is after I had finished my undergraduate years and had the good fortune of working in a lab, worked on the transparent part of the body. And when I usually ask audiences, do you know what the transparent part, the most, is it the largest transparent part of the body is? About 3 to 5% of the audience will raise their hand. Most of the people go, oh, what's the transparent part of the body?
Hmm.
And I then say, okay, I'm going to give you a hint. The hint is it's right in front of your eyes because it's the cornea. And it is literally right in front of your eyes. But I have essentially never met anyone other than these people who I had the good fortune of working with, who actually were asking the very interesting question: how is it that this part of our eye, how is it that that's transparent? What is the structure or what is the mechanism that allows it to be transparent? You know, if you have a misfortune of scratching your cornea, it becomes cloudy, somewhat like the white of the eye. And the cornea itself is in part clear because it has a very interesting structure to it that allows light to penetrate through. But it also has a certain amount of hydration. And when you scratch the cornea, that hydration changes and it becomes opaque, and people have to have corneal transplants. But virtually no one that I've met has ever asked themselves the question, I wonder why the cornea is a clear tissue, is a transparent tissue that allows me to see. And I think this is a fascinating question, but there are many more of these questions that arise in daily life. So I think that these are things that people looking for problems, it's not the big, fancy, complicated problems. There's a wonderful science fair for high school students called the International Science and Engineering Fair, ISIF. It's the culminating science fair for science fairs from all over the world, and people do come from all over the world. Almost 2,000 or so students every year, and I go to these periodically. And I'm going to tell you about one that is in this same light. That is one of my favorites. It was a student named Abigail Freed. And I was walking among the displays and I happened to see her. And so I went up to her and I asked her about her project. And when I talked to her, she started off describing her science project in a way that I don't suspect I'm ever going to hear again. Her first sentence was something on the order of, well, the thing you should know about me is I'm a ballerina. Not what you expect science stories to start with. But then she told me that not only was she very good at ballet, she was almost at a professional level. And professional ballet dancers have a real problem. And that problem was the one she decided to work on. What was the problem? It turns out that ballet shoes have a sole that's made out of cardboard. And because of the flexibility that's needed in the shoe. But after a single performance, that sole is no good and has to be replaced. Actually, the shoe has to be replaced. So these professional grade dancers use a pair of shoes every performance. As a result, it's very expensive. And so her project was simply to come up with a different type of material to be the sole of a ballet shoe. And she was able to find that. It was a carbon composite material that she used. And I thought that was a great idea, seeing a problem that was right around her and of her interest that she could follow. The second thing is that we have lots of assumptions about how we think the world works. You can find those assumptions, as a friend of mine, Mu Ming Pu, is a fantastic neurobiologist, does, by simply going to a textbook. Because what's in the textbook are all the things we believe are true. And for years, Mu Ming did a wonderful set of experiments that were all based initially on going to a textbook, seeing a hypothesis in the textbook or a result in the textbook, and then simply asking the question, how do we know this? What's the evidence? And usually you'd find that there wasn't a lot of evidence for the idea. And he'd say, well, I could retest this idea now. And he did. If the experiment confirmed what had previously been written, he'd simply say, well, I can say now that I've confirmed it with a more modern experiment. Many times, however, he was able to prove that that idea was wrong, and he now had another wonderful paper on how he now had new evidence for a different idea. And so I think this double approach, looking at what's around us and questioning our assumptions, is very important. For me, I think the third thing is when I read things, I'm always thinking about the experiments that I'm involved in. And so they trigger new ideas. So starting something triggers something else that then triggers another thing.
So, in your entire professional life, if you had to give one lesson, one piece of wisdom that you think would be most beneficial for the youth of today, what would that be?
So I can tell you what I would not want to hear, but I hear it all too often when people suggest things to students. What they say to students often is, follow your passion. I don't think that's the right thing. For a number of reasons, but one of the reasons, if anyone ever told me when I was in high school or college that I should follow my passion, I would get a panicked look on my face and say, wait, I don't know what my passion is. Tell me what it is. I don't want to make a mistake. I don't think the vast majority of us ever know what our passion is. I don't think that's the approach. But I want to change the phrase, and I do think a different approach is maybe important. And that is, don't follow your passion because you don't know and you're going to change your ideas and you're going to change what you do a lot. That's okay. But whatever you do do it passionately. Go into it wholeheartedly. It may not be your life work. It undoubtedly won't be your life's work. But if you do it enthusiastically and become 100% committed to doing it, that's a wonderful approach to it. Don't do it half-heartedly. So that's my only real advice. I wish I had heard it myself more to pay attention to myself. But that's what I think now is uh, a good approach to things. Be involved, be committed. So I want to argue against what you said. I don't know. I have a different view about what the Nobel is. The Nobel, I mean, I think everybody that works in science wants their experiments to succeed. I think they want things to be worthwhile. The Nobel is a very unusual prize in my mind. It does not go to the smartest scientist. It doesn't go to the scientist that has the most funding or the largest lab or writes the most papers. What it goes to is science that has either changed the way we do science, GFP is a good example of that, or how we think about science, how we think about the world around us. And often those types of discoveries, changes, are not things that are planned. They're not observations or something that I'm going to discover X. That often doesn't happen. But what happens is that someone will be doing an experiment and it won't work. Or what doesn't work exactly the way you think it should. Or there's an unusual aspect about it that you'd never considered before. And fortunately enough, for the people that have won these prizes, they've had a chance to sort of stop and say, I wonder what that means, or I wonder about this. I'll give you a couple of examples. Arno Penzias and Robert Wilson were two physicists, astrophysicists, at Bell Laboratories. And particularly Robert Wilson was a little disappointed because his PhD project didn't work out the way he wanted it to. And at Bell Laboratories, there was a 30-foot microwave telescope. And he wanted to use that telescope to sort of revisit some of the problems he was thinking about as a PhD student. And when he did this, when the two of them did this, they needed first to make sure the machine was working. So they pointed it at the Milky Way. And they got a wonderful signal. Worked. It gave them a signal. But they were careful. And they wanted to make sure when it was not pointing towards the Milky Way, that it was zero. Because that's what the machine was supposed to do. So they pointed away from the Milky Way. It never is zero. They can never get it to go to zero. They can't. So their experiment never started. They're trying to figure out, why is it that it's not going to zero? Maybe it's not going to zero because it's a 30-foot microwave telescope. Maybe there are birds nesting in it, or maybe it's near electrical wires, or maybe it's airplanes going to the nearby airport that are interfering with the signal. Maybe they eliminate all the possibilities, and then they realize that maybe it's not zero because they're actually measuring something. And what they're measuring is what's called the background radiation of the cosmos. And it is the first real evidence for the Big Bang. And that's why they win their Nobel Prize. Other people, I'll tell a story in a couple of minutes about Shimomura. His discovery of GFP was completely by accident. In fact, the physicist Enrico Fermi another Nobel Prize winner, has a very interesting phrase. It goes something on the order of, if you do an experiment and it confirms your hypothesis, then you've made a measurement. But if it doesn't confirm your hypothesis, then you've made a discovery. So it's these unexpected things that often then lead people to look in new areas or look in new ways at things. And so the Nobel recognizes those new things, but also recognizes them not the day that they're discovered or the year that they're discovered, but did they have an influence on how we think or how we do science? And so it to me, it's a prize for the science, because that science has to be something that many people have been influenced by and affected by, not is it a good discovery or not. So it's the influence, the impact of what has been discovered that's important. So let me talk a little bit about green fluorescent protein. The first thing I want to present, which you've already sort of hinted at, is three sort of problems in biology. So the first one of the three problems is, how can we actually see, particularly in a living animal, particular cells? And the animal we work on in my lab is a small roundworm called Cenorhabditis elegans. It looks like, but we'd like not to see the whole animal. We want to see just these particular cells. These are particular types of nerve cells that actually are the cells that sense touch. You might ask, how do you test for touch sensitivity in an animal that's only a 25th of an inch long, 1 millimeter? And the way we do it is we use a very sophisticated tool. We take an eyebrow hair and we glue it to a toothpick and we tickle the animal either near the head or near the tail, and it moves away. And we look for mutants that don't do that. Touch them with the hair and they don't move. That's a good idea, a good candidate for it being a mutant animal that can't sense touch. Dead animals also don't sense touch, so we have to actually make sure they can move and respond to other stimuli, but they don't respond to the touch of a hair. And we have hundreds of these. But while this nice diagram shows you where those cells are, when we look in the animal under the microscope, this is what we see. You can't see those cells. You don't know where they are. You don't know if they're defective or if they are in the right place or anything about them, because we have no way of looking at them. So that's one problem. How can we see particular cells? Another problem is the problem of what happens over time. If we look and take images of cells, for example, over time, we might get a whole hodgepodge of images, all of which look different. But in fact, these are all stages of a cell dividing. But how do we know what the right order is? How do we know how these go together and how much time is involved between them? And the pattern turns out to be something like this. But how do we go and actually see that pattern taking place? And the third problem I want to address is, where are genes active? We know that people have something on the order of 20,000 genes. So do these worms. But not every cell activates every gene. We have skin cells and liver cells and muscle cells and cells in our corneas and our eyes. And all of these cells, bone cells, all of these cells make some things in common, but many things that are not in common. So all cells have the same DNA, but they don't always have the same active genes. And so we'd like to know, what are the active genes? Where is a gene active in the cell? And so these are the sorts of problems we were addressing in the lab. We were able to answer them in many cases. But then I heard about this man, Osamu Shimomura, and his work on the jellyfish. And his work on the jellyfish was really fascinating. He was trying to answer a fundamental biological problem. And that problem was, how is it that certain organisms can actually generate light? Fireflies, glowworms, fungi, bacteria, fish, all sorts of different organisms, including this jellyfish, can actually generate light. So it's a wonderful basic question. How is that happening? What's the chemistry, the biochemistry behind generating light? And it turns out that every different type of organism does this in a completely different way. So if you understand fireflies, then you just understand fireflies. You don't understand the other organisms. You got to study the other organisms. And so he set about to try to understand why this jellyfish could produce this beautiful green light. And when he did those experiments, he worked all summer on this experiment, and absolutely nothing worked. He was a very good biochemist, very accomplished scientist. Nothing worked. He would grind up jellyfish. He bribed his kids to find more jellyfish that he could then study, and nothing worked. One night, he's working late into the night. The experiment has failed again, and he decides he's going to finally go home for dinner. It's already dark outside, and he takes the failed experiment, the samples from the failed experiment, and he throws them away in the sink. The sink had some seawater in it, probably some other things, jellyfish parts, who knows what else was there, but certainly had some seawater. He turns off the lights, and he's about to leave when he happens to look back at the sink. And when he does that, the sink is glowing brightly, something he had never seen before. So that unexpected surprise made him think, what was in the seawater that allowed the light to be made that he had never added to his experiments before? He knew what seawater was made of. And the one thing that it was in seawater that was not in his preparations was calcium ions. And so he repeats the experiment, and sure enough, every time he squirts in a solution with calcium ions, he gets a spark of light, a flash of light. And he uses that, the ability for calcium to generate light, as the way to purify the protein. He purifies the protein and names it after the jellyfish. He calls it a quarren. And so he's actually solved his problem. His problem was, how does this animal produce light? You take the protein quarren, you add calcium to it, and you get light. But it was that throwing the prep in the sink that gave him the insight. Sometimes people throw things on the lab bench or on the floor. You never know what works. In any case, once he had this answer, he realized he had a second problem, because the light that was produced was the wrong color. It was blue light. The jellyfish were green. So was there another protein that if he added calcium to it, it would make green light? And he was just looking at the wrong thing? And he tried that. Nothing else worked like that. And so then he reasoned, well, maybe this light is what's produced. This is the light generator. But maybe there's something else that converts the light from blue light to green light. And so he took a lamp with blue light and he looked at all of his samples. And as he looked at all of his samples, he discovers, yes, there's a different one than the one with the protein of corn in it. And whenever blue light is on it, he sees green light coming back. In 1962, he writes about this molecule in a footnote. So you don't have to write a lot to get a Nobel Prize. You can just write a footnote if it's an important footnote. And his important footnote said, I found this other protein. I'm going to call it the green protein. And if you shine blue light on it, you see green. Well, we don't call it the green protein. We call it the green fluorescent protein, or GFP. And GFP is something that you add, that is there in the jellyfish, and it converts the light produced by a coryn from blue to green. No one had anticipated that there was a fluorescent protein. No one even thought there would be these things. So again, two accidental discoveries. One, that calcium was important, and the second, that there was a protein that changed the color of the light from blue to green. We can use this to actually answer that question of where is a gene active. So I want to talk a little bit about this. Again, here's this animal that we have that can't really see where the cells are or where the genes are turned on in particular cells. And to remind everyone, the simplest way of looking at how we make proteins is DNA is transcribed into RNA, and that RNA, we say, is translated into protein. And if we think about genes in general, the sort of main components of the DNA, genes basically have two parts to them. One part I'm going to call the product part. It's the part that says this is what should be made. This is the RNA that should be made, or and then ultimately the protein that's going to be made. We sometimes call this the coding sequence. But there's another part to the gene as well. And this part we can call the control region. Because not only does the gene have to say what should be made, but it also has to say where, when, and how much of it should be made. So there's both a control region and a product region. Now, when I first heard about GFP, realized this is an incredible molecule. You shine blue light on it, you get green light out. And there's a picture of what the molecule looks like. And it looks exactly like it should. It's one of the few molecules in the world that actually looks like it's supposed to. It's a cylinder with a single part, it's called an alpha helix, down in the center, and that's where the light's produced. So it really does exactly look like a lantern, and that's how it functions. It produces light when you shine blue light on it. And I realized what a great tool this would be. What we could do is we could take that control region, and instead of making the normal product, we could just have it make GFP. And that will tell us where and when the gene is active. We'll see which cells make it because they will be the ones that turn green. And to give you an example, and one that allows us to see the particular nerve cells we like, is this picture here. So that's same animal or the similar animal, but now only looking at the cells we're interested in because those are the cells that turn on this particular gene. And that gene is now turning on the production of GFP. We can also make another type of fusion, and that is to take the whole gene and then add GFP at the end of it. So now the protein is really a hybrid. The first part is the normal protein that's made, and then the second part is GFP. So we call the first of these reporters a transcriptional reporter because it says something about the control of the gene. But if we make a translational reporter, where we now make the protein attached to GFP, we can then follow that protein wherever it happens to go. And so we can watch the protein. And this is a different sort of question, or set of questions that we want to ask. So this is where we can ask, where is the protein? Lots of advantages of using GFP over other methods. First of all, it's a protein. That means it's encoded by DNA. And if it's in the DNA of an organism, that organism can pass it on to its progeny and their progeny and so on. So it's not do an experiment once and then have to repeat it all the time. You can make an organism, an animal, a plant, a bacteria that has GFP in it, and all of its descendants will also make GFP because they all have the gene for that particular thing. You don't need anything else. You don't have to add any other chemicals. You don't have to play with the tissue. The only thing you have to do is add blue light, and you can see green. Doesn't hurt the animals to shine blue light on them. So we're not doing something that's really going to be disruptive or damaging to them. And it's a small molecule. It's only 238 amino acids for the protein. It's very compact because it's in that cylinder form. And that means it goes everywhere in the cell, and you can see it everywhere in the cell. And so it allows people to see cells in their entirety. Another component is, of course, because it's a protein, it's not detrimental to the organism. We can actually see this in living cells on living organisms. And it can be seen almost anywhere. So we put it in bacteria first, and then we put it in worms, and other people put it into flies and zebrafish and mice. And to give you an example of where it goes everywhere in a cell, this is a cell. I'm going to show you a picture of a cell from the brain. It's called a Purkinje cell. And you can see how GFP has gone everywhere in this cell. So you can see all this elaborate arborization and really get a wonderful idea about what the cell actually looks like. But as I said, one of the really wonderful aspects about this is to be able to watch living tissue. And I'm going to show you two quick movies, both about fruit fly development. And they were both done by someone I've never met, but she put these films on a repository, and I've really enjoyed them. Rosalind Silverman Gavrilla. And what they're going to show is nuclear division. Now, in the early development of fruit flies, the embryo doesn't have cell boundaries. It just has nuclei, and the nuclei divide over time. So it doesn't exactly look like this, but this is approximately it. You start off with a nucleus, and eventually the nucleus breaks down. The chromosomes are separated on what's called a spindle, and they separate to the two halves, and two nuclei are formed. So I'm going to show you two movies that she made. The first one has GFP attached to a protein that's part of the spindle. We're going to watch are 4 rounds of nuclei dividing in an early fruit fly embryo. The movie is sped up, so you don't have to take a half an hour to watch it, but you're going to see 4 rounds of the nuclei dividing. So here they go. Now the spindles form, they unform. They're going to form a second time here, a third time, and then one final time, fourth time, and then it's going to loop around.
Wow.
You should see it. There you go. And one of the really striking things about this movie is that everything seems to happen at the same time, that there's a coordination, and being able to watch things in living tissue really introduces that question. You want to know, how are all of these divisions taking place at the same time? And scientists do know a lot of information about this. But that's by using something that's part of the spindle. I want to show you her second movie. And this movie looks at GFP in the nuclei in the early embryo. And here, what you're seeing is a movie that's falsely colored. The more GFP, the more towards the red part. So now you see all the nuclei. The nuclei break down, and now the GFP is everywhere in the cell. And then coalesces again and goes back into the nuclei when the nuclei reform. So if there's no nuclei, it falls apart and it's everywhere. But then once the nuclei form again, it's able to be sucked into the nuclei because the GFP is attached to a small peptide that brings it into the nucleus. The first movie she calls In Synchrony. This movie she names after the Van Gogh painting Starry Night. And again, it's false colored. The more you have, the more you're in the red part of the rainbow. So what can you do with this? Well, let me give you some examples, or one example. We've used it in my lab to look for variants in cell development. So normally in the top part of the figure, you see that triangle, and that triangle points to the cell body of a nerve cell. And you see that that nerve cell sends out a nice thin little process that goes off to the left and nothing that goes off to the right. We say that these cells are monopolar. They go in one direction. But we were able to find a mutant that's shown in the bottom panel. And now the cell body is not at the end of the nerve cell. It has its normal part to the left, but now it has a part to the right as well. It's now a bipolar cell. That is, a cell with 2 processes growing out of it. And so we would never have seen this or never have detected it by looking for touch insensitive animals, because that's not what would happen. We actually had to look at the cell and see that it was different. And we've used this, and other people have used lots and lots of experiments to, in fact, look at all sorts of variation. You can look for changes in the number of cells, the position of cells, the outgrowth of cells, how they're connected, all sorts of things once you're able to see it. So if you can see something, you can study it. And I think that's the main lesson of GFP, is that it allowed people to see much more than they had previously. A couple more examples, and then I think I should be done. And the first example I want to give you is one that was a question in, one would say, human health. And that there's a movie of a mouse cell that's infected with the AIDS virus, HIV, and you can see a little viral particle being transferred right there to the adjacent cell. The reason you could see it is because the virus had incorporated within it the gene for GFP. So whenever there was a transfer of the virus, you'd be able to see it as you do in this picture. Why is this important? Well, you could see normally when we teach how viruses go from one cell to another, we often talk about bacterial viruses. And these viruses, a cell explodes. It opens up and releases lots of virus that then attack other cells. Well, if it's free and out there, maybe you could have a vaccine, antibodies against the virus that would scarf it up. But this says something different. The cell wasn't exploding. The cell transferred the particle from the infected cell, the green cell, to the non-infected cell. No explosion, no release. So now the question is not, how do we get something to sort of vacuum up the virus particles so they don't work? How do we prevent the cells from interacting to exchange this material? So being able to watch it happen, I think, is a very useful thing. There are a couple of researchers in Japan that decided to put the genes not only for green fluorescent protein, but red fluorescent protein and blue fluorescent protein into and connect them to the protein that goes into the silk fiber. And they were able to generate silkworms that could make fluorescent silk. And as you can see here, they even made some clothing out of the fluorescent silk that they made. I'm not so sure this is as useful as one might imagine, because I think you have to constantly be in an area where you can shine an ultraviolet light on yourself to show people how interesting your clothes are. Another example, which I quite like, is by a guy named Bob Berledge. And Bob Berledge knew that there were some genes that had been found in bacteria that could be activated by the explosive TNT. And so he decided to make some bacteria that would become fluorescent. They turn on the production of GFP when those bacteria were in the presence of TNT. So why would he do something like this? Well, he actually knew that landmines, which are one of the really horrible aspects of war, leak TNT. And they injure non-combatants and people well after any conflict has left an area, because not all those landmines are discovered and removed. And he wanted a way of discovering landmines. And so what this picture shows is a 3-meter by 5-meter plot of land that he had a friend bury 5 landmines. These were disconnected. They weren't going to explode, but they did have TNT in them. He sprayed his bacteria on the ground, and then came back in the evening and took this picture with fluorescent light. So he was looking to see, could he detect the presence of landmines? So hopefully they wouldn't hurt anybody. Now, the problem with this experiment is what we call the problem of false negatives. If you say something isn't there, and it really is, that's a false negative. And we don't want that, especially with landmines. And so people are still working on getting this to be an accurate method that virtually works every time, or it works a surprising amount of the time. They want to get it to 100%. People are still working on this. And then 2 other examples from the world of art. The first is a Brazilian artist named Eduardo Koch commissioned a French biotech company to make a rabbit that expressed GFP in its skin. This was his pet rabbit at home, the GFP bunny, Alba. And Eduardo Koch would bring this animal to his art shows to get people to talk about the connection between art and technology and art and science. And you and your fellow students may be wondering, has GFP ever been put into humans? And the answer to that is that there was one claim of what we would call a transgenic human with GFP several years ago. And I will show you a picture of this person. He's right there. And in the movie Hulk, it actually starts off with the jellyfish swimming around, and a hypodermic comes in and takes apparently the GFP out of the jellyfish. And that's how Bruce Banner gets his green skin. Somehow it is incorporated in some gene that gets expressed in skin when he gets angry. And I actually know the screenwriter for this movie. He's here at Columbia with me, and his daughter and my daughter went to elementary school together many years ago. And I went to him, his name is James Shamus, and I went to him and I said, James, I saw your movie. I really liked the beginning. How did you know about my research? And he looked at me and he said, I have no idea what you work on. And I said, oh, it's that GFP that you had at the beginning of the movie. How did you put that in? And he said, oh, we had a student from MIT who was working on this, and he convinced us that that would be a good idea. So GFP has made it into the movies.